Hello, good morning. Ooh, got some responses on that. Thank you. A lovely crowd for 8:30 on a Friday. Welcome, everybody. We're going to get started here and kick off this great day. For those of you who I haven't met yet, my name is Brittany Ritchie. I work here at Seattle Children's Research Institute and Research Institute Administration, and we are pleased to be hosting today's fair in partnership with Northwest Rare Disease Coalition. So thank you all, and so many of you who've also taken the time to be here today. So this is our first time holding this event in person in nearly 4 years. How many of you were here in 2019 when we had it in a different building? Anybody? No, a fresh crowd. Okay, maybe some folks on WebEx were here in person last time. So thanks all for making the time to attend in person or virtually, those of you who are online. We have the privilege and pleasure of partnering with Carolina and her team and many of you to bring this event to life because all of your passion and purpose aligns with our mission, which is to help kids live a healthier and more fulfilled life around the world and locally here in Seattle as well as across the country. So by the nature of the diseases that affect children and the portion of the population, we consider most pediatric diseases to be rare diseases anyway. So we are happy to be a part of this event for the, fourth time or something like that, fifth time over time here? Yeah, 2016, I think. So great. So with a few exceptions, most of today's event will be recorded and shared out afterwards for those speakers who have consented to do that. So you'll have the ability to go back and review later. And those of you who can and are interested, we hope you choose to join the Rare Disease Summit tomorrow, which will be entirely virtual. You can enjoy that from the comfort of wherever you are. So I'll touch on a few housekeeping things to set you up for success, those of you who are virtually especially, and anyone in the room here as well who's maybe logging in online as well. So first of all, we've got a couple QR codes here. These are placed around the space rather than sacrificing a bunch of trees. We've got these to save some paper, so feel free and scan those now. I'll leave them up for a second, or they are on some stanchions around the space, or you can go to the Rare Disease Fair website to find them yourself and see all of the great things happening and the awesome people making it happen. Okay, so first I wanted to let you know a little bit about who we are in case you're not familiar with Seattle Children's. We've been a part of the Seattle community since 1907 as a clinical care facility, providing care to children regardless of their ability to pay. We are, our research division itself, which is where you are now, is about 17 years old and is closely connected to the hospital, which really gives us that strength of that clinical and research connection to make advances. So both are supported by our amazing foundation and our supportive community, which we could not do this work. So together we all work towards that ultimate mission of providing hope, care, and cures to help every child live the healthiest and most fulfilling life possible. At the research division, that means leading or supporting research of all types in a wide range of fields, from the impact of media use on children's development, screen time, all that stuff, to the groundbreaking cancer immunotherapy treatments being developed on the 12th floor of this building, Cure itself, and dozens of projects related to SARS-CoV-2, COVID-19, gene editing, all that sort of stuff. So in a variety of fields. So we have a wide breadth of work. And we're proud to be a part of this event where you're all putting your heads together to move in that same direction to help kids live more healthy and fulfilled lives. So if you're online, this is the housekeeping bit. If you're online, I suggest you are muted by default. We suggest you open your chat windows. That's where you'll put your questions in and someone in the room will read them out loud. You can also choose to raise your hand if you're online during Q&A and we'll unmute you. You can't do that on your own because of how we've set up the event. If you do have any issues online, just leave the event and come back in using the same link. Close down other applications or chat to Rachel in the chat box. And she is our WebEx guru and amazing event planner colleague who's in the front row. Hi, Rachel. I'm going to embarrass you. Thanks for your support. Now, without further ado, into Carolina Summer. Thank you.
Is this the? Okay, awesome. Thank you all for coming here to the 2023 Rare Disease Fair. So happy to have everyone here. I just can't believe that we're back in person again. It's been a while. It's just so good to see people and everyone. And I just want to say that I'm so grateful, we've been doing this fair, I just want to say, since 2017. So it's just really exciting to see how much it has grown. So grateful for our partnerships with Seattle Children's Research Institute. for hosting this event. We couldn't do this without you guys. A million thanks to Brittany and Rachel and the AV team here at Seattle Children's, and for your wonderful collaboration. And Amy, thank you for taking the load off my shoulders this year and stepping up. And she makes the most beautiful spreadsheets. Just love it. So grateful for her work. And then thank you to Joshua and Max here too. They're the co-founder. Sorry, I'm trying to figure this out. All right, thank you so much to Joshua and Max too they're also the co-founders of the Northwest Rare Disease Coalition. And Joshua is our industry person and Max is our policy person. And together, it's just been a wonderful collaboration. Thank you. But the fair is really a total team effort, you guys. And it's really this whole community coming together. And that's one of the things I love about this fair is that it's a lot of us making this work. And it's all of us here who are passionate to help this rare disease community. We have over 50 speakers between today and tomorrow, amazing speakers, and including some youth. And I just love seeing and empowering the new generation of advocates. And it's just so great to see them. It's gonna be a great day, you guys. I'm excited. A couple of things, there's a photo booth by the elevators. You can kind of get the map and the QR code for the agenda and for the bios. And then I think the codes are in the registration and up front here. There'll be some educational videos going on by the photo booth, but also visit our Stories Are X, and they have a booth as well, and they'll be doing some small videos on people's experiences receiving a diagnosis. A couple of things. So there's a couple of little things. Oh, upstairs there's the kids area. So we have a lot of activities like tattoos and face painting and all kinds of fun and story time for the kids. And then in the hallway, we have a rare artist here, Malachi, and I'll be introducing him in a little bit here. And he's done some amazing artwork that you can purchase too. So we're so excited to have him. If you're joining us virtually again, just please put your questions in the chat. And so now I get to kind of introduce myself. My name is Carolina Sommer. I'm the CEO and founder of Born a Hero Research Foundation. I'm also the co-founder of the Northwest Rare Disease Coalition, the founder of the Seattle Rare Disease Fair, and most importantly, I'm a rare disease mom to Mariana. My daughter just had 2 surgeries 2 days ago. So, she's 11 and has had 12 surgeries. The hard times come with tears, maybe little soft cuss words here and there during urgent times, and also a lot of chocolate. But for us, brain surgeries never get easier, and we have learned to cherish every moment and every day that we get to be with our children. We do not take any day for granted. For many patients living in the world where it takes 6 to 9 years to be diagnosed is crazy, and we have advocates here. We had an advocate lobby with us that took 50 years to be diagnosed, and it was weird to me to tell him that that was a pretty big deal to take that long. So this world of the unknown is something that we can all really relate to with all that's happened with COVID, right? Just the world of the unknowns, and this is just kind of what it's like for rare disease patients on some level. So let's just say that more than 1 in 10 people have a rare disease because it really affects entire families. I have been sick since my daughter was born. I have been taken on an ambulance with my daughter at some point because it was too much that day for my heart. So, and it was sorry. Okay. I was in the ER last week with several health issues, hoping to feel good enough for her surgeries 2 days ago. Okay, and so here we all are still advocating because it's just part of our life, and I'm not alone. Sarah here was lobbying with us from the hospital, and Sabrina went lobbying with us after her son just had surgery. But so I just, these women are total super women, and I just wish they didn't have to carry such a heavy cross. At the same time, I just want to say, there's so much to be grateful for. Here's a story. I visited a village. I just want to say a quick story. We visited a village 3 weeks ago, actually. We just came back from Panama. These people lived on an island. We were on an island 45 minutes away from the shore. And but really they made their own power boats. That's how they travel. So it took more than 45 minutes to get to shore for these villages. And I can tell you this, but then to get to the nearest hospital, it was about 4 hours from shore. And I share this because it's important to also be thankful for the gifts that we often take for granted. Our son had sugar level dropped while we were on the island, of course, and we had no medication, like Parents of the Year, right? We'll never do that again. But it was a miracle because we were able to keep him safe and stable all the way home through the boat ride and through the bumpy roller coaster rides all the way 4 hours to a nearest hospital. And I just kept thinking, this indigenous people, and my heart just breaks for them because it feels like they don't really have a choice like we do in all these, like hospitals like we have. So anyways, with that being said, thanks to all the wonderful doctors, the nurses, the researchers that save our lives every day. Thank you to all our industry and biotech companies, because without you, I know at least my daughter, my son, and I wouldn't be here. And thank you to our legislators because of their hard work. Last year, legislative session, I know they had more than 1,000 bills that they had to consider, and during a really short session. So it's tough just making simple decisions. They have to make really hard decisions. And here's my mommy part of the speech, I guess, in the end of that. We have to be grateful, be kind, and collaborate to accelerate research, you guys. There's good and bad collaborators wherever we go, but most people have the best intentions and want to help. We can't take sides and assume things about people and parties. We have to work together and include everyone to make change. Patients, industry, biotech, payers, physicians, and scientists all working together. We're in this together, and we can really benefit from each other and be stronger together. We just need to find a common ground and start from there. So thank you for my wonderful husband and kids, and for all your support. And again, I want to say, I haven't even moved this. I was just so into here's my family. Took some pictures of our trip and stuff. And then I think, and then here, I just want to say thank you to our sponsors, because we really couldn't do all this without you and your support. Seattle Children's Research Institute, Pfizer, Pharma, Illumina, Vertex, Ultragenics, Alexion, Mozi Therapeutics, Regeneron, Desmond Consulting Groups, Born to Hear Research Foundation, and the Northwest Rare Disease Coalition. And then we're going to show a quick video, an intro from Vertex, really quick. Do I need to do anything for that? Or? I just keep going. Okay. And then just, okay. Oh no, okay, see, I messed it up. Okay, do I need to go back? And is something supposed to happen? Okay. Something is going to be happening in a second here, I guess. I don't have to do anything, right? No. Okay. All right. Thank you. I don't know when I'm going to cry ever, so it started early this time. Usually I wait till the end. But yeah, just get it out of the way. I'm better. Is he waiting? Okay. Suspense is really great. A really great video. I guess in the meantime, while we wait, is it ready? Oh. It's not showing. I can start introducing Malachi Castillote. He will, Malachi. What am I saying, Malachi? Okay, I don't know. Malachi. Thank you. I love you, Sabrina. I'm going to introduce Malachi. Oh, okay, here's the video. Thank you. Malachi, I'm sorry.
Hello, my name is Marissa Dunn, and I am thrilled to join the Seattle Rare Disease Fair and Summit on behalf of Vertex Pharmaceuticals. I would like to begin by thanking Carolina and the Northwest Rare Disease Coalition team for hosting this wonderful event, and most importantly, all of you for engaging in the important topics and discussions throughout the summit. One of the main reasons I joined Vertex was the passion I felt from every employee to pursue science with the aim of transforming lives for people with serious diseases. Fast forward to today, and our commitment to patient communities has not only grown stronger, but is the guiding light for who we are as a company. By putting patients at the heart of everything we do, we've discovered and developed medicines for cystic fibrosis, a rare life-threatening genetic disease. Beyond CF, we are focusing on other rare diseases, several of which disproportionately affect people from ethnic and racial minority groups, including sickle cell disease, beta thalassemia, and APOL1-mediated kidney disease. Our goal is to develop treatments to address these devastating diseases, and we won't stop until we reach that goal. The Seattle Rare Disease Fair is one of my favorite events of the year because it brings together all areas of the patient advocacy community. Today, you will hear from expert speakers and take part in panel discussions that will highlight best practices for how life science companies can incorporate the patient voice into the work they do. These conversations will show the importance of advocacy and engagement to help improve the rare disease community's experience, which is something that is extremely important to us here at Vertex. I want to thank all of you again for being here, and I look forward to seeing the outcomes of this summit, and I hope to see you around today. Thanks.
Thank you so much, Marissa. I don't know where you are, but thank you. And I'd like to introduce Malachi Castillote. He will give a quick talk and introduce the moderator for another neurological rare disease section of the conference today. Oh, there he is. Okay. He's a teen advocate with Batten disease and an artist. Art is a way that he expresses himself. So we're just I'm so delighted to meet him and honored to have the opportunity to introduce him.
Feel it. I'm in school. Yeah. Hi, I'm Malachi Castillote. I am in the Hello, I'm Malachi Castillote. I am the artist that's out probably roaming in the halls when we're all done here. I am a CNL2 Batten's disease patient at Children's Hospital. Currently, I am the oldest at 22 years old, still talking, still walking, and still fighting. I'm going to talk about a little bit about my art. So I always tell people that I don't have, people always ask me if I have an inspiration before I do an art piece, and I always tell them that it's abstract. I don't have an inspiration. I just pick colors and put them on canvas and see what comes out. So all my canvases are just what I just came up with on the fly. That's pretty much it. But my art is getting a little bit harder because I am partially colorblind, and there's certain colors that I love to use more than others because I can see them a lot better, like blue. So I have so many blue canvases in my house. I like the color blue. You said
Sorry. He's like, read what? Say what? Thank you, Malachi. I'd like to introduce the next moderator, Ellen Morgan. She's the founder and president of PROS Foundation. That's correct.
Could I just ask a question? Is the artwork for sale?
Yes.
Well, great to see everybody in person. I haven't been coming to this for 5 years, but I did attend virtually last year, so it's really nice to be back in person again. I'll be moderating the neurological disease section and want to introduce our first speaker, Dr. Rami Akilin, who is joining us all the way from Israel. So we really appreciate him making the trip here. He is a full professor of immunology and cancer research and serves as the head of the Institute for Medical Research, Israel, Canada. His research interest is to study how unstable genomes affect disease development, especially its cancer development and neurological disorders. Of particular interest for his group is studying the developmental epileptic encephalopathies using mouse models in brain organoids. The lab has a major focus in translating bench-to-bed work by developing gene therapy approaches to tackle rare genetic diseases. So welcome, Dr. Akilin.
Good morning, everybody. Thank you, Ellen, and thank you, Carolina and Effie, for really the invitation. It's really an honor to be here. And sorry for the mess. You can see that I'm still on jet lag, and but awake, I should say now. So I'm going to tell you today about what we're doing in the lab, and I want to introduce how we come to be more interested in neurodevelopmental diseases, and give a focus a little bit on two genes, actually, the history of what we're doing with WOX and the CTNNB gene. But before diving in, I just want to remind everyone that rare genetic diseases are not as rare as we think. When I approach this subject, it's a lot of patients are suffering from rare genetic diseases. In the US, 30 million patients are suffering from rare genetic diseases. In the world, there is more than 350 million patients with rare genetic diseases. This would form the third populous country in the world. And many of these about 7,000 rare genetic diseases, they are affecting mostly children. And among these, the majority actually affect the neurological system or the nervous system. I assigned here the years because this is just to remind me and tell you that I am in training a cancer biologist. And in 2015, I started this journey studying neurodevelopmental disorder because of having to know and to see families with children that are having a neurodevelopmental disease, a very devastating, that I will tell about a little bit. So as I said, I'm a cancer biologist. We are interested in studying, okay. You can see this? Yeah, maybe this is better for the Zoom. We study these, what we call common fragile sites. These are fragile regions in the genome that harbor genes that are important in cancer. And these, we call them tumor suppressor genes. Okay, and this is the prototype that we use. It is a gene called WOX, W-W-O-X. And we studied it intensively. And as I said, in 2015, it was discovered that this gene is altered, is mutated in a very devastating epileptic encephalopathy disorder that was named after WOX as WORI, for WOX-related encephalopathy, epileptic encephalopathy. And it is an autosomal recessive disease where the two the two alleles are affected. Ultra rare, we know of maybe 150 to 200 cases in the world today. And the children are really suffering from seizures, ataxia, visual intellectual disability, vision problems, very complicated, and they don't respond. They are refractory to the anti-epileptic drugs. The major problem, I would say, that differentiating between Worry syndrome, which is the severe form of the disease, to SCAR12, which is a milder form of the disease, is that Worry syndrome children have an average of 1 to 4 years old to survive. Whereas SCAR12, they probably survive, but they are ataxic and they're in very bad shape still. So we wanted to see what we can do, because we intensively researched the WOX gene. And what my lab is doing is really to take and make models to be able to study the significance of the genes, the basic mechanisms of how these genes would really result in a phenotype like the one that I described. So we did generate mouse models that mimic what happens in the patients. Others have generated rat models. We also did work on cells taken from the patients to reprogram them and to be able to better understand what is really the phenotype, what is going wrong with these cells coming from the patient when they are lacking the WOX gene, for example, in this case. And more recently, we've been also generating, I'll tell you a little bit more about this, what we know as brain organoids. These are three-dimensional structures that we grow in the lab, in the dish, that try to mimic those brain development that is happening in the brain of the children. With this understanding and this modeling, our aim is really to develop therapeutics. Okay, and one focus that my lab is doing is gene therapy, where we wanted to restore the expression of genes into the brain of the affected children, hoping that this would normalize their phenotypes. And we did, as I will show you briefly, that we did a proof of concept that could really provide a good therapeutic venue for Worry children and SCAR12 children. But now we're moving to develop this further into the clinic. So just a word about gene therapy, you probably know, but we have a cell that has an affected gene, a mutated gene. Usually, these are genes that are lost. They display loss of function, so they are not functioning normally. Okay? And what we want to do is to bring this gene, this intact normal gene, into these cells, replacing, oops, I don't know why it just keeps going, replacing the affected mutant gene, and this way hoping that the cells now will function normally. So this is what we refer to gene therapy. So we're adding a copy of the DNA of the gene, the normal gene, to replace those unfunctional, abnormal gene. So in essence, what we did for WOX gene therapy is that we generated this vector, what we call a vehicle that is carrying the WOX gene and driving its expression very specifically in neurons. This is why we use a specific promoters that called synapsin 1 here. And the vehicle that would bring this into the cell, into the neuron, we choose to be an adeno-associated virus. This is a virus called flu virus that probably most of us have exposed to it. And we take all the harmful elements from it and only leave it as a vehicle to transfer and to get our gene of interest into the cell. And this way we take advantage of this viral, unharmful virus to transfer or to bring the normal gene. Sorry. The same strategy we're doing with CTNNB, which I'll talk briefly. But again, we are replacing, you saw here, the WOX gene. Here, we just replace the gene with the CTNNB, the beta-catenin, and hoping that we can bring an extra copy of this defective gene into the neurons and normalize the function of the neurons. So just to wrap this up, so we have identified that there is a malfunction of a gene in a specific disease, right? This is what you do in the genetic testing. And then you generate a viral vector to harbor this gene of interest, and you want to bring it back. You check it in animals. You generate animals. You need the models, animals or brain organoids, and then you phenotype these animals or models that you generated when you restore the gene, the normal gene, and hope that this will normalize those cells and those animals. Okay, so I will show you just a couple of data here that we did with the WOX gene, because I think this is remarkable and impressive. The mice that are null for WOX, they are smaller in size and they die by 3 weeks of age, okay? And when we treated them with the AAV9 synapsin WOX, this is the gene therapy for WOX, these mice grow normally, they gain weight, and they survive already for 1.5 years even. So this was a proof of concept that we are very happy with, because we could show that WOX is indeed responsible for the disease. If you restore its normal function, you can rescue these mice, these mutant mice. Another interesting thing that we observed, which brought us very close to the CTNNB field, is that when we generated brain organoids from the patients, I'll explain more about these brain organoids, one thing that we noticed is that there is hyperactivation, there is increased activation of beta-catenin and the Wnt pathway, and this was happening in the knockout, and what you see in the violet is that when you restore WOX, when you try to rescue, you can normalize these genes, at least most of them. So again, another model, another platform can also support some mechanistic understanding of gene function. And this was very critical for what we know as the regionalization of the cortex. Our cortex in the brain, this region in the forebrain, is set in layers. So we can really look at the different layers. We can see that in the knockout, this is similar for mouse and human, is impaired. For example, you can see that this SETB2 marker is almost not present in the knockout, but when you restore WOX now with the AAV, you can restore the layering and the normal layering. And why was this very interesting? Because this is what we see also in postmortem patient samples taken from Wari children. So this was shown by others, but it is very supportive to what we did. So this is the roadmap. So we want to do modeling of the disease using animals, using brain organoids, and using these models in order to translate and now brings in some therapeutics that can help the patients. And I'm very proud to say that our technology has been adopted by Mazi Therapeutics. I think most of you know Yael Weiss. She's taking this forward to the clinic, and we are very excited that this is moving forward, and hopefully we'll have great news in the coming few years. Okay, so with this very general introduction, at the end of 2021, I met Lital. Lital is a mom for a CTNNB child, Itamar, and we were in the news, so she saw the reports, and she came to meet me, and she described me what is happening with her child. And we started really looking more in depth into the CTNNB syndrome, about the gene, about its loss of function, about the clinical phenotypes. And this is what we learned and we know that children with CTNNB syndrome, where there is a loss of only one allele, in this case, has intellectual disability phenotypes. They have developmental delay. They don't walk, they don't talk, they don't see, or there's vision problems. And some of them have also microcephaly and cerebral palsy. So knowing more about this, so there was this very recent article showing the patients, 404 individuals with the CTNNB variants. And we learned that most of the mutations that are affecting the CTNNB gene are those that have a truncated gene, meaning that we have a immature gene that is not being that is produced, affecting really the normal function of CTNNB1 and beta-catenin, or others that have frame shift where the reading of the gene is impaired. So again, affecting the normal expression of the gene. And what was really very striking is that all of those patients with the variants, most of them, they have really very common and similar phenotypes, as you can see here with the cognition, movement, speech, and so on and so forth. The gene is very critical gene when we talk about CTNNB, encoding this protein that is known as beta-catenin. It binds so many other proteins and affects so many functions. What is critical that I want just briefly to mention here, that this is part of a pathway, a very conserved pathway in worms, in insects, in mammals. Very important pathway. The downstream effector of this whole pathway is beta-catenin, encoded by CTNNB1 gene, and it is acting as a co-activator for genes in the nucleus, in the that gives the order for the cell. And what I want you to pay attention here is that there are several important components here. Let's just take this GSK3 alpha beta enzyme, which really it phosphorylates the beta-catenin, the protein, and then inactivate it, okay? And then resulting in so many different functions. Some of these functions are important for neurogenesis, the production of the neurons, but also for other cell types in the brain. And there are two main functions that I want to emphasize here for CTNNB in the brain. One, that it is regulating the proliferation of the cells, so how much cells we have, and to what different cell types they differentiate, they become. Okay, and this would affect their function if they are a neuron or supportive cells like astrocytes or oligodendrocytes, different cell types in the brain. So it affects proliferation, the number, but it also affects the structure. So you need this structure of the neuron, something that is maintained by what we refer to as cytoskeleton. It is the cell skeleton that is shaping the interaction and communication between the neurons, what we call as synaptic structures. And these are the main two functions that I would want to emphasize here. And you can imagine if you have a problem with CTNNB and a mutation and it is not functioning well, then you are affecting the number of the cells. Probably this is why you got also microcephaly, and the cytoskeleton of the cell, and this is why the neurons are not communicating and networking in good way. So we wanted to study this more in depth, and these are the problems when we face in any rare genetic disease. There's rarity of subjects. Animal models do not necessarily always phenocopy what happens in the human. You can think about 10 million neurons in the mouse brain, whereas we have 10 billion in the human brain, so 1,000 more. And of course, we cannot always work on post-mortem tissues, so we need to develop new models, and this is was the focus that we want. So we wanted to take cells from the patients and put them in the right context. Beta-catenin and CTNNB has been intensively studied in cancer because when it is overexpressed, it is leading to cancer. Here we're talking about loss of CTNNB, and we wanted to study this in the neurons, not in any other cell type. And this is why we use the patient-derived stem cells that I will talk briefly. Okay. So this is what is the great advantage. We can take cells from the patients, we can reprogram them, tell them to go back in history, become a stem cell, and then differentiate them to very specific cell type or to change them by genetic editing, and then really study their function by different functional assays or by studying considering different technologies. Organoids are those three-dimensional structures that we can generate from these cells, from the stem cells, and because of the fact that they can form different cell type within these structures or this organoid structure. And they were very much shown to mimic or to be phenocopying what happened during human brain development. This is why we focus on brain organoids. So what we did, I'll summarize this in 2 minutes. Sorry for the delay. So we got blood samples from 2 CTNNB patients in Tel Aviv. Both of them, or one of them has stop codon. Again, the mature protein is not produced from one allele, and the other one has a frame shift. So again, it is at the C terminus, however, which might be very interesting for the results. We produced stem cells from these, induced pluripotent stem cells. We validated these cells. We wanted to make sure that they go with the quality controls of karyotype, so their chromosomes are normal. They have markers that are really showing their intactness. And then we started to look at what is happening to the beta-catenin and some of its downstream targets. I want to show you here that there are changes of subtle changes, and probably this is because the mutations are at the C terminus. We wanted to generate the organoids, and we specifically choose a very specific protocol, and this is what we can see here in the picture. When we grow these structures in the lab, in the Petri dish, this is how they look like. This is from last week. And in order to test now some drugs and some therapeutic options, we collaborated with Dr. Michelle Jacob at Tufts and Michelle Viewer at MIT, who generated these JSK3 inhibitors. Do you remember those that inhibit beta-catenin? And this is the one slide before last. And you can see that in the patients, we can increase this green staining around the cell membrane of these 2 patients, and not in these, for example, treatment, because these are different drugs. And this is the same for another gene that affects the structure. You increase here the staining, not in the other treatments that are controlled. So I can say that we are hoping that with this brain organoid setup that we are now expanding, we can study more the mechanism of CTNNB defect in the neurons, but also try different therapeutic modalities, including small molecules and drugs, but also gene therapy that we are doing, and hopefully provide some proof of concept in order to take this forward to treat CTNNB patients. These are the guys who did the work. Raul is now taking over, and this is the group. Thank you very much to you for listening, and to our collaborators and the funding. So questions at the end, right? Yes. Thank you.
So thank you so much. So next we have Dr. Guillermo Cerati, and he is a native of Argentina, He received his medical degree in Buenos Aires, and then he came to the US in 2003 to do his residency in pediatrics, and then followed up with a fellowship in molecular genetics and cancer genetics. He joined BioMarin in 2015 as regional medical lead covering all BioMarin commercialized products in Latin America. And then in 2019, he joined the U.S. medical team, where he has been serving as Senior Medical Director for CLN2 and MPS. Welcome.
Thank you, Ellen. Thank you, Amy, and thank you, Carolina, for inviting me and the organizers. It's a pleasure for me to be here. And let me see if I can go to my presentation. There we go. So it's a pleasure for me to be talking today to all of you about CLN2 disease, which is an inborn error of metabolism resulting in neurodegeneration. And as it was mentioned, I'm the Senior Medical Director for CLN2 in BioMarin within the US. Let me see how to advance. There we go. So as you all know, neurodegenerative disorders of childhood are complex and debilitating conditions. Usually they are multisystemic, difficult to recognize by clinicians, and they have a difficulty in diagnosing and managing these conditions due to their rare nature. And most commonly, they are caused by a genetic disorder. In a report, said 63 to 75% of intellectual disability and neurological deterioration cases were caused by an inborn error of metabolism. And we all know that timely management is key to preventing brain dysfunction and function decline. We usually start with a steady state, and when we have a neurodegenerative disorder, there's the neuronal damage and clinical symptoms that, if not treated and or recognized, can create irreversible neural damage that we cannot resolve as in other organs and tissues. And inborn errors of metabolisms are a group of rare genetic conditions which have as an underlying etiology disorders of metabolism and are amongst the most common genetic causes of neurodegeneration in childhood. These conditions are caused by genetic mutation, typically monogenic with autosomal recessive inheritance that cause a deficiency either on the enzyme or transporter of an enzyme, typically in a metabolic pathway, and with the consequent pathologic interruption of a metabolic pathway leading to toxic accumulation of materials in different organs and tissues, or the inability to generate or transport energy-carrying compounds. Diseases, if we take all inborn errors of metabolism together, are highly heterogeneous and in presentation and disease course, and is highly dependent on the pathway that is blocked. The last international classification for inborn errors of metabolic disorders accounted for 1,450 disorders described in 24 categories. So you see there are a lot. Individually, each one of these are very rare, but collectively they affect a large number of individuals worldwide. And we see some reports in which the overall global birth prevalence of all inborn errors of metabolism is high, is 50.9 per 100 live births, which is roughly 1 every 2,000 births. And in areas where there's higher incidence and prevalence due to inbreeding, like Middle East and the Mediterranean, this goes even higher, 75.7 every 100 live births, with roughly 1 in every 1,350 births. Which means that, for example, in Turkey, in a report, they showed that they have roughly 840 children who are born with an inborn error of metabolism every year. And inborn errors of metabolism frequently have neurological symptoms and developmental disorders. Although very heterogeneous, 80% of them have some form of neurological manifestation leading to encephalopathy. as manifested by seizures, epilepsy, and developmental decline. Here we see that developmental and epileptic encephalopathy, apart from the developmental impairment that occurs due to the genetic etiology, also added has the effect of the excessive epileptic activity that affects each one of these conditions. And one of these inborn errors of metabolism, one of the groups is the lysosomal storage disorders, which are an heterogeneous group of 50 disorders caused by inborn errors of lysosomal pathway genes, resulting in either enzyme or protein loss of function. Here we see that either enzyme or cofactors of enzymes or membrane transported proteins of these enzymes are defective, leading to the progressive accumulation of material in the lysosomes. And many times diseases are classified based on the aggregate substrate. And as examples, we have the mucopolysaccharidosis, the oligosaccharidosis, and the sphingolipidosis. But these lead to deterioration, cellular and tissue function, and they are multisystemic, affecting the musculoskeletal, GI, cardiac organs, and also cause neurodegeneration, as we mentioned before. An even subgroup of LSDs is the NCLs, which are the neuronal cell lipofuscinosis, which are a group of conditions that have in common a common pathophysiology, which is the accumulation of this autofluorescent material, in particularly affecting the neuronal and retinal cells, although it has to be highlighted that the accumulation is multisystemic. That's why in ancient years, skin biopsies done in patients was the only way to diagnose. So you could see the accumulation happens all over the body. They share similar clinical characteristics, but they vary in age and onset and disease course for each one of the forms of NCL. As NCL progresses, the classical debilitating symptoms that they manifest with are vision loss, motor dysfunction, epilepsy, dementia, and they are the leading cause of dementia in children and adolescents. That's why it's so important to diagnose them timely. CLN2 disease, which is the condition that I'm going to be talking more in detail today, is also known as tripeptidyl peptidase 1 deficiency, or TPP1 for short, and is one of the 14 identified NCLs. It's a rare and rapidly progressive childhood neurodegenerative disorder, and is one of the most common forms of the 14 described NCLs. There's an autosomal recessive inheritance of pathogenic variants in the TPP1 gene that creates a loss or reduced enzyme activity of this TPP1 enzyme, and that particularly some tissues are very sensitive to this loss and the accumulation of these autofluorescent materials, and these are the neuronal and retinal cells. And this creates progressive disease manifestation with seizure, epilepsy, dementia, motor deterioration, vision loss, and premature death. And here we see Noah Van Houten, who is a patient, very well known, who passed away from CLN2 disease. Motor and language difficulties are some of the most common first reported symptoms in CLN2 disease, and we are fortunate that Europe collected data on CLN2 patients for the natural history, and here we see that the first noted reported symptom was seizure. That's what brings families with the kids to the ER and to care. And in this database, seizure onset was between 2 to 4 years of age, and it included a vast variety of types of seizures: myoclonic, tonic, tonic-clonic, absence, but also what you see there following seizures, language difficulty, which might not be the first identified by the families and the first identified by clinicians, was already happening even before seizures, and is often missed. Follow this is the motor difficulty that can be comprised of ataxic gait, clumsiness, tripping, and frequent falls. But here we see the natural history of CLN2 disease with a predictable and rapid decline of function. And this is very descriptive of one of the phenotypes of CLN2 disease, the late infantile form, which for which there is much more information. We know that there are other phenotypes that we're going to mention later on. And the dental cohort from Europe between 2002 and 2016 showed this rapid and predictable neurodegeneration through the CLN2 rating scale, which is a rating scale that has two main domains, the motor and the language domain. Each one is scored from 0 to 3, with a total combined score from 0 to 6, where lower scores represent more extensive impaired function. And what they showed is that whilst patients were first having the first seizure at around 3 years of age, with fairly preserved motor function, but already language delay, they were really diagnosed 22.7 months later, were at around 4.5 years of age, where already they were not able to walk without assistance, and their speech was hardly not understandable. So this shows that a delay in diagnosing this patient poses a large burden on the patient due to the rapid progression of the disease. By 6 years of age, many of these patients, due to the cumulative progression and the cumulative signs and symptoms that these patients have, are severely impaired, and they become very heavily dependent on their caregivers. They rely, because they have dysphagia, they become blind, they become bedridden, they have this cognitive decline and loss of language that makes social interaction very difficult. And also the myoclonus and the seizures become resistant to drug treatment, so it becomes very difficult to manage. And these patients and their families require a lot of support. But as I mentioned, I just described the classic late infantile form, which is the most common form of CLN2 disease, with this slow in development, psychomotor regression, language delay, epilepsy between 2 to 4 years of age, the retinal degeneration that and for which patients are blind at around 5 to 6 years of age, and a shortened life expectancy between 6 to early teens. But as opposed to this, other forms of CLN2 disease called atypical vary in onset, progression, and disease manifestations. They do not always start, and there are several reports that do not always start with a seizure as the late infantile forms. They might start with behavioral problems, autistic spectrum disorder manifestations, movement disorders, and ataxia. And we've seen patients identified through not seizure panels, but ataxia panels. But it is important to note that regardless of the phenotype, there's neurodegeneration and premature death. And there's an overlap in the patient management and support needs between different phenotypes of CLN2 disease. And that's why it's so important to do an early genetic testing to avoid that delay. Genetic causes underlie a substantial portion of genetic epilepsies. And nowadays, pediatric neurologists are very aware and they're very familiar to performing molecular genetic testing through panels, which allows to have very low suspicion clinically wise. They do not need to have a particular suspicion for a condition, and just send a panel or even a whole exome sequencing. And in the case of CLN2, there's need to detect 2 pathogenic variants, one of each allele of the TPP1 gene. But with panels and with the amount of information that comes back with them, and more even with the whole exome sequencing, there's the challenge of variant interpretation and the burden of the variants of unknown significance and what to do with when this happens, what to do with the information we received. So ideally, and for whenever it's possible, like for CLN2, and this molecular testing should be paired with some form of biochemical test. In the case of CLN2, with a TPP1 enzyme testing to assess the level of enzyme activity in patient samples. That requires as well to have a clear diagnosis and teasing out the level and making sure that the sample is of good quality and appropriate control enzyme. But again, it's very important to do timely diagnosis. And through some reports from panels, we've seen that there has been progression in shortening the time to diagnosis of patients when they start having symptoms. This requires not only familiarity of the specialties with genetic testing. I'm a geneticist, so I'm very familiar and very comfortable in ordering genetic tests, but other specialties who are not genetics, sometimes they are very hesitant, particularly with the information they get back with a report. And it is important for the early diagnosis, early intervention, and potential outcomes for not only CLN2 patients, but all patients who have a genetic epilepsy. In this, the benefits are confirming, of course, the diagnosis, have a very specific and a name for the genetic epilepsy we're in front of. Of course, shortening the diagnosis, these reports have shown that they were able to shorten diagnosis by 1 to 2 years, which we've seen it's critical for CLN2. We saw the slope of decline that these patients had. So doing an earlier diagnosis, we are able to catch the patients who are not as affected. Also, it allows, and this is my genetic side, for specific counseling, particularly family counseling, to inform about the type of disease they're in front of, the type of inheritance, what that implies for the rest of the family, and also put in place very specific targeted management. If there's no particular specific management, it would allow potentially to participate in clinical trials and also avoid unnecessary testing that sometimes some patients are subjected to, limiting the overall diagnostic cost. For CLN2 disease, as some of you might know, there have been published assessment and management guidelines that sort of guide on the baseline and ongoing regular multi-systemic assessment, such as something very physical, like something very basic, like the physical exam, including general health, growth, vital signs, visual performance, frequency of seizures with a seizure diary, that's very important for this patient, the developmental assessment, language and motor difficulties as rated by the CLN2 rating scale. It's important to keep that scale assessed on a frequent basis. The feeding difficulties, if the patients start having that, to put interventions in place. And also medical and medication utilization. We want to make sure that they are not overly medicated and that the medication that they use is very specific and efficient. Also, as you guys know, these patients lose sight. They need age-appropriate ophthalmologic evaluation and eventually OTC to assess the macular and retinal degeneration, as well as neurological epilepsy record and ciliary testing with MRIs to look at the volumetric, the volume of the cortical gray matter to assess for loss, and also ongoing EEGs to characterize other types of seizures that would be amenable to different anti-epileptic drugs. Holistic care is a big word for these patients, and I'm almost done. And it's key to support the patient and the family. And when I talk about the patient and the family, I'm not talking just the parents and the patient itself. I'm always very aware of siblings who might not be affected, but psychologically they are very, very concerned and sometimes neglected by the amount of attention and energy that CLN2 patients require. So a pediatric neurologist, rare disease specialist with clinical experience in CLN2 disease, supported by a multidisciplinary team, should lead patient care. And here we see the pediatric neurologist supported by a nurse, occupational, physical, speech therapists, movement disorder experts, the ophthalmologist, as we mentioned, the geneticists. But as disease progresses, also they need dietitians, social workers, and counselors in place. And to finish, it is important, and I wouldn't stress more, the holistic care that is critical for CLN2 management. And the multidisciplinary care is advised whenever possible. These patients are complex to manage, and it requires the collaboration of multiple HCPs in constant communication with the patient's family to make sure that the patients are able to obtain the most of the specific management that they're going to get into and improve outcomes for patients with CLN2. And with this, I thank you, and I will be open for questions.
Thank you very much. I wanted to mention that at the end of the neurological section, there will be time for questions and answers for all of the speakers in this session. So next, I'm really happy to introduce Sabrina Castiote, who is a wife and mother of 3 young adults. She's had a successful career as a corporate executive administrator in managing conventions for the Washington State Convention Center. She spent 3 years deep diving into all aspects of CLN2, trying to save and maintain the lives of 2 of her 3 teenagers who were diagnosed with a rare variant. She's been an advocate for helping her kids and other rare disease kids. Thank you, Sabrina.
Good morning. Okay, I'm going to try to put my big old laptop up here real quick. Thank you guys for having me. Well, thank you for BioMarin speaking. I'm like listening to my whole life story all just right there on the screen and everything that he said. I'm like, yep, sounds about right. All right, I'm not a speaker, so I'm going to try to get through this. So here are a couple things that I'd like to touch on, but I can't touch on all of these things. So I'm just going to kind of jump in and dive into how our kids got diagnosed. Our diagnosis took us 16 years plus to get diagnosed. Malia was deemed failure to thrive her entire life, and she had relied on a feeding tube. Her bilirubin was 28.9 at birth and landed her in the ICU for over 5 days. Fast forward, she completely fell off the growth chart entirely at 8 months, never knowing that this was actually related to a rare disease that was stealing her life. We thought we were just feeding her for growth and not trying to keep her alive. To be clear, we were denied genetic testing at the age of 3 due to insurance. Malachi's story is a little bit different. So this would be my second child that has been diagnosed with CLN2. Malachi's story was started about 8th grade when we noticed that he started having tremors. And after being diagnosed with autism at the age of 14, he started wearing glasses a lot. We had to change his glasses often. A lot of migraines, vision changes occurred randomly. There are times where he would pass out in the shower. We did not know why. Never knowing why, it wasn't until his sophomore year in high school, after being struck by a vehicle while riding his bike to school, did we finally get to a concussion team, but also a constant persistence on my end of wanting a neurologist. That neurologist at Seattle Children's recognized my true desire to help Malachi, but also it's finding that doctor who's willing to go the extra mile. As Malachi's, we learned that his cerebellum was actually shrinking and it was not normal. And reviewing his past history and details, why he had tremors, why they were progressive, only on his left side of his body, but after the accident transferred to the right side of his body, we were finally granted genetic testing. Weeks went by, and what we thought we were arriving at was just a regular follow-up appointment. It was different, as when the team of doctors and social workers filled the room, I knew something was off. But also thinking Seattle Children's is kind of a teaching hospital as well, so it's pretty common for them to ask, hey, can we have this person in the room? So I'm always like all for it. But the staff started to fill up quite largely in the room, and then when I saw a social worker or when she presented herself as a social worker, I knew something was different. The news was actually given to us not of Batten CLN2, but actually kind of an old term used SCAR7. Even at that moment, I realized and knew and understood more than the team that had come in. And that was because me and my husband, when we did a deep dive into SCAR7 before the official diagnosis, which actually led us to Batten's disease. Sorry. Which we didn't actually think it applied to our kids. He was 18. This, what we were reading, was actually applied to infants and toddlers. It all became a blur after that, and requests were granted to test our 2 other kids the following day. And we spent an entire week sharing with our kids and Malia and our son Azrael, who our middle son is, that Malachi would be going through brain surgery, that we needed to travel to another hospital out of state, and that he had a rare disease. A week later, we received a call from Seattle Children's Genetics Counseling to inform us not only did Malachi have Batten's disease CLN2, but our only daughter also, too, came back as a rare disease CLN2 patient. I can tell you that day, I truly lost my hearing for hours on end after that phone call. It is amazing how your body instantly takes on all this anxiety, and it took it on to a whole nother level that I had never experienced. Malia was about to turn 14 a week later. We decided to wait an entire week and a half before we'd share this news with her. And as we shared this news, that brain surgery would be in her future, her entire body shook with fear. I would just say that just tears literally just flowed from Malia, and all we could do was literally hold her super tight without ourselves breaking down. We had already spent a week and a half hiding this from her. Life changed in an instant. We had a week and a half to pack and pull our life and their life out of school, sports, activities, literally flown out of state to Orange County Children's Hospital, where we lived at the Ronald McDonald House for about 4 months. It was a huge challenge to pay for everything, including our home at home and being away from home, and doing our current, now to our current situation. The thing is when you first get diagnosed, fundraisers literally just pop off. It's wonderful. People jump in, you're blown away about how people care and give. Even Make-A-Wish jumps in, except for when you're 6 months out of turning 18 and you're denied a Make-A-Wish trip. That was Malachi. We'll talk about that later. That's for another story. I can go on a rant about that. But here we are, and we're at this stage where it's, how can you help me make a life through this journey? All the amazing support truly gives us this illusion, the idea and feeling that we're okay. We'll be able to manage this. Look at all the support we're receiving. It's actually quite deceiving. Listen, the private jets, that's cool. That's really cool. It's fun, but it was actually donated. So it helped us cope with the cost of flying. We were flying every 9 to 10 days, bi-weekly, every other week for 6 months to Orange County Children's Hospital. So these jets were donated by people who own jets. But we didn't get this all the time. We didn't have a jet every time. We actually flew commercial, and we didn't always have the funds to do it. We finally actually end up switching to driving across 3 states for medical care, medical follow-ups, and being a part of a study. Examples about organizations. I would say that organizations, we've heard lots of partnerships with organizations, and one that we've actually learned and heard about was Games for Love. They actually provide resources and gaming systems inside the hospital. And because my kids were older, video games were cool. So they wheeled these things in. The problem with that was Malachi was also older. They only allowed you to play games that were for little kids, not for older kids. And so, you know, they're in the hospitals, they help kids with medical crisis, but this organization has realized and needed and understood the resources that we actually need long-time care, and that would be in our hospitals during the summer, but at home during the school year, after they graduate, to maintain their terminal disease. I would say we are definitely beyond grateful for people who have started this journey with us. They can kind of snap on that bracelet, you know, that honor of I've helped. But we're kind of past that now. We're in this transition where we need the t-shirt of I'm still helping. People start to fall off after a while. And you start to come to terms with the rare disease, that this isn't a one-time event. You're good, in a couple months you're good. This is a long-term progressive disease, and it's terminal, and you will not be, it's basically lifetime. After months and years, people have already given in their attention, but their attention fades away. And you begin to really understand the depths of the disease, everyday costs rolling in, more expensive, and generally that you didn't really anticipate. Because you already know the cost of treatment, but it comes to the care of treatment. With progressive rare symptoms get bigger, the impact gets greater, the day-to-day life activities begin to change, the cost and complications of day-to-day care increases. Small things like glasses, sorry, I'm a little time's up, are 12 pairs of glasses in 2 years. Tremors, dropping glasses, unable to use normal utensils, and then we switch to adaptive utensils. The cost of a fork maybe costs $2. Adaptive forks, $20. This cost is put on us as families. I'm unable to work. Let me describe the medication. The medication for our 2 kids is $174,000 per dose times 2 kids. That's $696,000 in a month. I would tell you that the constant fear that I sit in and thinking about and needing to keep applying for financial aid every 6 months is exhausting. I would say that the financial impacts for our family is that we had no choice to file for bankruptcy. We had to put our kids through 2 brain surgeries, signing our life away, our documents, losing our vehicles that we had spent our lifetime building to try to get to. The other cost that they don't tell you about is when you have surgery and they call you the night before and tell you that you owe a whopping $9,500 for both of your kids to have brain surgery. They've had brain surgery twice. They now have chest ports that lead directly to their brain. I'm definitely out of time, but I thank you. I haven't been able to finish the rest of my speech, but this is my family. Malia and Malachi are my Battens 2 CLN2 fighters. Their squad support is their brother, who also feels the impact of this, and it impacts him greatly. And then this is my husband and myself advocating for our kids. We need a cure. We've been battling with the FDA. We need a cure. We need gene therapy. Thank you.
Thank you so much, Sabrina. That was sad. The next part of this session is a panel discussion about neurological overgrowth diseases. Shauna, who's our first speaker, will provide an overview, and then Kim and Jill, who will be presenting virtually, will talk about the diseases they represent and the collaboration between them. So first I wanted to introduce you to Shauna, and I'm so appreciative of Shauna because she stepped in about 3 days ago to do this presentation since the original speaker had something come up and wasn't able to participate. Shauna is a board-certified genetic counselor with a specialty in neurogenetics. She has almost 20 years of experience in both research and clinical care for neurogenic conditions, and has helped to build multidisciplinary neurogenetic programs at several institutions where she provided genetic counseling services and coordinated the care of patients with rare neurological disorders. She's now at Seattle Children's Hospital, where she's helping to build a neurotherapeutics program. She's worked directly with patient advocacy groups and has been on medical advisory boards for several of these groups. So Shauna, thank you.
Thank you, and hopefully people are enjoying all the talks. I am enjoying them myself. So we're just going to do an overview of neurological overgrowth syndromes or diseases, and then a couple more panelists will talk about some specifics on types of these. So this is a large collection or a large variety of conditions that can cause, for example, discrepancies or differences in brain size, or differences in skeletal growth, or significant differences compared to the general population in size of these either organs or body systems. And as I mentioned, there's a huge collection of these types of conditions, all with many different genetic causes. And there's going to be a couple that are highlighted later. So like I said, these disorders are associated with several often severe coexisting symptoms depending on which systems are involved. Sometimes these disorders are very rare and may not be on physicians' radar. So often these conditions end up being underdiagnosed, particularly because, as some of the other speakers have mentioned, there is kind of a bottleneck when it comes to access to genetic testing. For example, getting in to see a provider who's familiar with ordering genetic testing, and then the cost or coverage for genetic testing can be a barrier, as well as interpreting genetic testing results and knowing what that means for families. So there's a lot of barriers when we're working with people to get them access to testing. And also, just like these diseases, there's many different diseases, there's also many different types of genetic tests. And so people, even people in the medical institutions, may not know what is the best test for my patient. These tests range in terms of like very small, like single gene tests, or they can go up to panels. Panels, which were mentioned earlier, are sort of a grouping of genes that can all, when there's changes or mutations, lead to a similar, what we call a phenotype or a symptom presentation. And so panels can be in various sizes, from 20 genes to 100 genes to 300 genes or more. And that's what we call phenotype-driven testing, looking at symptoms, clinical presentations, and selecting a panel that might identify the genetic cause. But there's differences in panels, even from lab to lab and things like that. So you could have a certain symptom presentation that a physician has appreciated and diagnosed, and you could have a panel done at one lab which did not find a cause. But if you select in a different lab, there may be a gene on that panel that actually would have found or identified the genetic cause of your symptoms. So even between different labs and things like that, there can be differences in these panels, and it may not be capturing everything that could be causing that person's symptoms. So we can sometimes miss people depending on what they're being tested for. So a lot of times people are now discussing things like exome sequencing. So that captures all the active, or what we call protein producing genes that a person has. So that is 20,000 genes. And sometimes we are also moving towards genomes, which is the exome is 1% of a genome. And so that we're talking about a lot more data. There's pros and cons to each of these approaches. And that's a lot of times what a genetic counselor does, is working with families, talking about the pros and cons. What's the best choice in that situation to get to an answer for families? And one of the concerns that a lot of providers have is, the more you test, the greater number of genes, for example, you test, the higher likelihood you are to get variants of uncertain significance. These are changes that may be unique to an individual or very rare in the population, and we don't know what it means. We don't know if it's actually associated with a clinical presentation or if it's just a benign change that's unique to that individual. So this is a huge area of kind of difficulty when doing genetic testing, and a lot of providers are not comfortable with dealing with variants of uncertain significance, which means it's very important when people are undergoing genetic testing to help us kind of report those variants, get them out there on public databases like ClinVar and things like that, so that we can have a history of finding that variant. And then as more and more people are found to have it, maybe comparing symptoms and finding out, does this really cause disease? So what I'm talking about in variants, and just to catch people up if they're not quite clear what a mutation or variant is, when we're talking about genetic information, our genes are structured on chromosomes, and genes are simply kind of represented by a code of letters, our genetic alphabet, which is very simple, a combination of A, C, T, and G, which are put together in three-letter genetic words that spell out to the cell what it's supposed to do and how it's supposed to do it. And it's creating, each of those three-letter words are coding for amino acids, which are then strung together like beads on a string that make the proteins that function in the body. And so these are proteins that are essential for kind of growth and development and all the things that our body is meant to do. So when we talk about changes in what we're trying to detect with genetic testing, it could be, for example, a single letter change, like a T that's been, something that's like a G should be there, but a T has replaced that. And then it translates to the cell to create an incorrect amino acid, which changes the function of the protein. But sometimes people have these spelling changes that are just unique to them, but they don't cause disease. And so it's our job to figure out, is this actually disease causing? Is this the cause of this person's symptoms? You can also have things like deletions, missing parts of that gene, or you can have like insertions or things that change that code in different ways that again lead to these abnormal proteins. And in some cases, in terms of neurological overgrowth syndromes, you can also have whole extra copies or missing copies of a gene. So you can have what we call a duplication or deletion of an entire gene that actually could lead to size discrepancies. Brain sizes or head sizes that are too large or too small. And this is just a list of examples of different genes. And you can see when under the title CNV, if these particular genes symptoms are caused by duplications, dupe, or deletions, dels. When we're talking about overgrowth syndromes, it's also important to know that there are just common variation in the human population. So these are changes that have to be statistically significant to be caught and lead to genetic testing and diagnosis. And so there's a variety of different things that are used, including neuroimaging and other testing, to guide us along the way as we're approaching genetic testing. I don't want to get too complex into some of these, but I know that we're talking about a lot of different conditions that are often very rare. Maybe only 200 or 300 people in the world have them. And people say, well, how do we get people interested in this condition? It's so rare. How do we get researchers here? But remember that, for example, in this indication for mTOR pathway, there are a lot of genes listed along the side that can be all involved in this pathway. So your sister group could be another gene that is important in the understanding of this whole process, and kind of collaborating with other organizations, other patient advocacy groups, and of course researchers is essential for this. I've been involved with a lot of different development of clinical trials, natural history studies, development of outcome measures, and it is vital to have patient and advocacies in partnership with clinical researchers to move that forward, to help us develop things that are important to the patient, not just things that we can measure in an exam room. And so I'll end there since I'm out of time, but I appreciate everyone. Thank you.
Thanks, Shauna. So our next two speakers will be presenting virtually, and I don't know exactly how this is going to work. The first one is, I think, is Jill going first? Yes. Okay. So our first speaker is Jill Kiernan, and she's the executive director and founder of the TBRS community. Her daughter Avery was one of the participants in the childhood overgrowth study that first identified TBRS. Jill and her team have built the community research network where researchers, clinicians, and families collaborate and work together to further patient-centered research toward treatment development.
Okay, can you hear me? Can you hear me? Okay. Hi, everybody. I'm Jill Kiernan. I'm the founder and executive director of the TBRS community. And if you can see my slides, this is my daughter, Avery. Just shout out if you can't. Avery, I noticed right from the start that she was not developing typically from birth. She was really floppy. Her head was large. She grew really quickly. She had a lot of feeding issues. I knew something was off. My pediatrician at the time didn't really listen. And finally, when she was about a year and a half, he sent us to the neurologist. We didn't have the best experience there based on her imaging and presentation. He told us that he thought that she had a disease that would cause her to die within 2 years. And this was the day before Thanksgiving, and Thanksgiving's never been the same since. And he told me to prepare myself, and very luckily she did not have that suspected disease. She did not die in 2 years. But as Avery grew, it was clear that she was not okay. She spent more time in the doctor's offices and therapy appointments by far than she ever played on the playground or with friends. After a really long diagnostic journey, at about 10 years old, Avery was one of the first 13 people to be diagnosed with Tatten-Brown-Raman syndrome as part of the childhood overgrowth study in the UK that first identified the syndrome in 2014. So we were relieved that we had answers, but didn't really have many answers. We were scared and alone with no information. I reached out to Dr. Tatten-Brown, and she was incredible. And we also started the Facebook group and started to connect with other families. And eventually we created a nonprofit organization called the TBRS Community in October of 2017. And our mission is to support all families affected by TBRS and to advance research toward interventions. So far, we have identified about 300 people globally with Tatten-Brown-Raman syndrome. It's caused by a variant in DNMT3A, and I'll preface my talk by saying I am not a scientist. I am a mom, I'm an educator, and ended up being an executive director because my daughter and all of her, she calls them syndrome friends, needed help. So the majority of our leadership are parents who came together when we were first learning our kids were diagnosed, and there were maybe 20 of us in the world with no answers. And never doubt the power of a rare disease parent. We all joined together, and from the start, we knew that people with TBRS have overgrowth. They're typically tall in stature, have increased weight, large head circumference, intellectual disability, and facial features like heavy horizontal eyebrows that become more apparent with age. And that's kind of all the information we had at first. So we launched the TBRS Community Patient Registry, which is a natural history study, and we used NORD's platform. And that has helped us to learn more about TBRS. So we know now that it's linked to autism. 50% of our patients have cardiac abnormalities. 50% have had at least one known seizure, and about 20% of our patients are diagnosed with epilepsy. Unfortunately, our children have an increased risk of developing leukemia and other blood cancers. 83% of our patients have some mental health diagnosis, and also many have orthopedic and mobility concerns, neurological issues, sleep apnea, regression, vision problems, difficulty with temperature regulation, GI issues, scoliosis, and the list goes on from there. So our core value at the TBRS community is the patient and families come before everything else. They are the reason we're here, and they drive our work. So it's important to us that every year we do a patient priority survey, and we survey our patients and families, and we create our programming around their needs, and we fund and recruit research in their areas of priority. So not surprisingly, their research and treatment development priority areas were centered around intellectual disability, mental health concerns, communication issues, and the physical disabilities that many of our families are challenged with. And they also, our families want surveillance and treatment guidelines. And 99% of people surveyed said that they would consider participation in a clinical trial. We have an incredible medical and scientific advisory committee to help direct our research program and to provide clinical information and research updates to our patient community. Most of these clinicians and researchers have come to our family conference and met our families in person, and that's where I first saw the magic of hearing the patient's story and the patient's voice. So over and over from the doctors who attended, I heard they were so inspired to go back to the lab and clinic after meeting our kids, and they found a new purpose for their work. And it also really helped inform their research agenda. So for example, one of our families during a Q&A mentioned that their child had regressed, and then 3 or 4 other hands came up, and out of 15 people there, and their children had regressed as well. And so then our researchers and clinicians went back to their labs and clinics and started focusing on finding answers for us about regression and TBRS, and that's still ongoing. In 2020, we developed the Patient-Led Collaborative Research Network, and that's a collection of clinicians and researchers from around the world who we encourage to collaborate to work towards treatment development. And we just started scouring the internet and looking for people who had published on TBRS or DNMT3A. There's a lot of interest in DNMT3A because changes in that gene are known to cause AML leukemia. So we kind of connected with everybody in that field. And, we just looked for synergies, and we were able to, one awesome example was Sir Adrian Bird, who is a prominent researcher in the Rett syndrome space, was our keynote presenter at our last Collaborative Research Network conference. And he spoke about the progress made in Rett syndrome and how that could potentially apply to the TBRS space. So our strategy has been to build our organization to advance patient-centered research, and we created our TBRS treatment toolkit to offer tools to help further research. So for patient data, we launched our community patient registry, which is a natural history study. We recently partnered with Citizen to collect electronic health records and data that way. And then we also launched our IRB-approved Just kind of simple patient directory, quick and easy, called Count Me In for TBRS, where we collect demographic and variant information, and it's recontactable in case of research and clinical trials that we want to inform people about. And we have a biorepository. I have a feeling I'm going over, so I'll be quick. Where we're collecting blood and urine, saliva, cerebral spinal fluid, tissue like skin and postmortem brain samples and tumor tissues. Sorry. And we're funding work on biomarkers. We have mouse models that recapitulate the syndrome, iPSC lines, isogenic controls, and fibroblasts. So we're really trying to amass everything we can to help move research forward. And our future goals are to hire a science director to help us develop a strategy that includes how to use those tools to move ahead. We're launching an EEG repository, working on developing biomarkers, assays, outcome measures, and hopefully eventually treatments. So I'm going to turn it over to Kimmy here, and I'll be back to talk about our collaboration together for the Overgrowth Syndrome Alliance. Should I stop sharing while you introduce Kimmy?
Sure. Just quickly, Kim is an MS in both special and general education and has dedicated the last 17 years teaching the special needs population. Upon receiving her son's diagnosis of Milan syndrome in 2017, she co-founded the Milan Syndrome Foundation, where she currently serves as president. I will
you want to just share your screen and I can just take it from here?
Yes, yes. Sorry.
It's all good.
I told you I wasn't good at this.
Sorry, guys. We're over here on the East Coast.
Okay. Oh, I'm not sharing, am I? Sorry. I just have to click this button.
That's okay.
Okay.
Okay, so hi guys, I'm Kim Venturola. I am the co-founder and president of the Milan Syndrome Foundation. A little bit about Milan. You can go to the next slide, Jill. I'll run through it quick. A little bit about Milan syndrome. It is a neurodevelopmental overgrowth disorder caused by de novo variants in the NF1X gene. The NF1X protein transcription is a transcription factor, meaning it can be found widely throughout the body, and it affects every part from head to toe. It is a haploinsufficiency, and the first patient was diagnosed in 2010. At this time, the first symptoms and markers are macrocephaly, so an enlarged head, either at birth or before the age of 1, hypotonia, and global developmental delay early in infancy. Recommended testing for an accurate diagnosis would be whole exome sequence or targeted panels for specific symptoms. And currently NF1X is on the macrocephaly overgrowth panel, the autism intellectual delay panel, and hypotonia panel, which is why we recommend whole exome sequence, because that's not nearly enough. We really are working hard to get NF1X included on other panels, for example, epilepsy, different vision panels, strabismus, nystagmus, optic atrophy panels. Next slide. So here I am. Like I introduced myself, I'm Kim Venturola, and first and foremost, I'm a mom. I have 2 boys. My oldest son is 12, and my youngest son is 8, who's pictured in this slide right here. That's Dominic. So Dominic is my child affected by Milan syndrome. And I have my master's in special education and general education. So I am in no way a scientist. So when Dominic was born, I was a special education teacher for early intervention. With Dominic, there was always something. We were constantly adding diagnosis after diagnosis. I knew something was going on. I didn't know what was going on. But I kept bringing it up. We got him into early intervention. He was 9 months old when we started, which to me was probably too late, but I'm grateful for what I got. And by 15 months old, Dominic had an entire team of therapists, every therapist you could have in EI, come into our home. We spent most of our time with that team. I took a leave from work to focus on Dominic's therapies and his care. I knew something was wrong, and I just had to figure out what it was. But I will tell you, and just like many other stories here, you're not ready for this world. I knew I wanted to be a special education teacher since I was a child. I never changed what I wanted to be. But to be on the other side of the fence and was truly a reality check and something I would have never imagined. But as you can see on my next slide, it really drove my journey forward. It's okay. So I was determined for answers. And like we were adding therapists, I was adding specialists. And we were adding specialist after specialist and diagnosis after diagnosis. We were going for second and third opinions. It wasn't enough. I didn't understand how so many things could be going on with one child and not have one answer. It didn't add up to me. So finally, I made a phone call to the Children's Hospital of Philadelphia's individualized genetic center, and I met with Dr. Azumi. And Dr. Azumi was honest. He said, listen, I truly believe Dominic has a genetic syndrome, but don't put all your eggs in this basket because there's only a 30% chance you will find an answer with our whole exome sequence. But we left there and we agreed to our whole exome sequence. We did put all of our eggs in that basket. And 8 weeks later, we did receive our phone call with an answer. And I can tell you, as I'm sure every parent that already presented is sitting here is listening to this with a child with a rare disease, you know the exact spot you were either standing in, sitting in, what you were doing, what you were wearing when you received that answer. It's like a gut punch, but for me it felt bittersweet. I finally had an answer. I had a reason. But then there was the bitter part. The reason was so rare. There were no answers. We were told there's a Facebook support group. There was about 4 publications and very little known. And believe it or not, half of my son's diagnoses, the geneticist told us weren't related to Milan syndrome, when in fact we know now that they were. So this picture here, I'm going to talk real quick, that of Rare Disease Day, that was our very first Rare Disease Day. My son was diagnosed 5 days before World Rare Disease Day in 2017. And that's a day I never thought I would celebrate or have an impact, that it would have such an impact on my life. But that is the day that we announced to the world of social media and family and friends that our son's life was ever changed by a diagnosis. Next slide. So what are the clinical features of Milan syndrome? It's very clear here. I'll be brief. Like I said, macrocephaly, tall and lean, overgrowth of the musculoskeletal system. You can see they have a morphinoid body style. They have distinct facial features. Hypotonia, intellectual disability from moderate to severe. They have severe anxiety, behavioral challenges, a lot of optic nerve issues and vision loss, hearing impairment. The list goes on, and we're constantly learning. We scour our patient registry. We push it so hard because we're driving the research. We are writing the story on Milan syndrome, and we're getting it out there. And the more we understand Milan syndrome and what it entails, the better we can help develop a standard of care. There are many red flags and markers. And if we can get those out there to the doctors and care teams, it can lead to quicker diagnoses for families. Next slide. Here's our timeline of progress. So the Milan Syndrome Foundation was co-founded by just 4 moms about 4 years ago. We all had various backgrounds. We had a special education teacher, a general education teacher, a neuroscientist, and a social worker, and we developed a very well-rounded organization. And for just being 4 years old, we've come really far. In just this short amount of time, we have a robust research program that was launched in June 2021. We have a biorepository. We developed our research roadmap with Prolera. We've partnered with Chords, Rare X, and Citizen. We listen to our patient community. We focus on the patient voice and the needs of our families to help support and raise awareness as best we can. Next slide. Next slide. Sorry, next slide. Can you hear me? Sorry. Oh, there we go. We have our Milan Syndrome Foundation contact registry, which currently has 216 individual families that we're in contact with that are genetically diagnosed with Milan syndrome across 32 countries. This is the only global contact registry for Milan syndrome. We know there's more out there. We just need to raise awareness and grow our numbers. Next slide. Trying to go as quick as possible. In January 2019, shortly after we were established, we created our website and we started our medical and scientific advisory board. These 6 individuals are absolutely incredible and help us drive all of our research. Any question we have, they're incredible to be in contact with families. It doesn't end with them. They're truly so passionate about Milan syndrome. Next slide. So this is our Milan Syndrome Foundation board. In August of 2021, we went from 4 moms on our board to a team of 11 international board members. We are all volunteer run, and every single one of us on the board are a parent or a grandparent of a child with Milan syndrome. In December of 2021, like I mentioned, we started our research network. We hosted our first Milan syndrome scientific roundtable. We had over 20 researchers involved. We continue to grow our research network. And at our family conference in July, we were able to host our second research roundtable. That was a hybrid event as well. Slide. And I think I saved the best slide for last. And just like Jill mentioned about the family conference, her family conference, we hosted our very first inaugural conference. In July, we had over 50 families join. So this is the first time that anyone has ever, that some families have met anyone else affected by Milan syndrome. We had our medical and scientific advisory board and our researchers there as well. And for those of you who have ever attended a conference for your child's disorder, you know the feeling and why this slide brings such a huge smile to my face, and why I saved it for last. The connections made here are absolutely incredible. What really stuck out to me is the collaboration, not just between the science and the medical side, but the collaboration between the researchers, doctors, and scientists with the families. We had doctors dancing with our children on the dance floor, geneticists swimming with them in the pool, researchers telling jokes at the young adult table, one of our top scientists pushing a child around in the luggage cart in the hotel lobby. That was my son. But these rare disease conferences are so meaningful. They're more meaningful than you could even imagine, because it literally does, like Jill was saying, it puts a face to a gene. It makes the research more relatable. It drives their passion and it gives hope. Thank you. We're going to go into collaborative slides. Jill, you want to just jump into it?
Okay, can you hear me now? I was stuck on mute.
Yes.
Sorry, before I was advancing them, but it wasn't showing that they were advancing.
That's okay.
Yeah, so our collaboration together with Milan Syndrome Foundation started when I reached out to Crystal, one of the founders, for help with our patient registry, because she had already launched one with Overgrowth, and I thought, oh, maybe she can help. I don't want to reinvent the wheel. and we were chatting, and she had not yet done a family conference, and we had. So it was a great partnership from the start. And for us rare disease organizations with very limited resources and not a lot of people to fundraise with, you don't need to reinvent the wheel. It's so important to share resources and knowledge and share your failures, especially because you learn the most from them.
Yep. So we were already collaborating, and the one thing through many discussions with TB, the Tatten-Brown-Raman syndrome, and a bunch of other overgrowth syndromes was we realized the overlap of symptoms was undeniable. So we really sat down and we knew we should be looking at this differently. We started raising questions. Could there be overlapping pathways between the syndromes? We knew coming together would help our patient communities ultimately lead to treatment developments for overgrowth syndromes. It was crucial and necessary to become more appealing to researchers. We also knew we had to keep the patient's voice centered at all times. And just looking on this screen, those are just six major overlapping symptoms, but there's so many more similarities than there are differences.
And that kind of led us to come together and create the Overgrowth Syndrome Alliance, or OSA. And we are bringing overgrowth, intellectual disability disorders together to collaborate and share resources and work toward treatment development, like Kimmy said. And just the first step that we see is coming back to the patients first, right? And understanding the collaborative voice of all of our overgrowth intellectual disability patients is so vital. So we're creating a shared collaborative patient priority survey, and we'll bring that collective voice to the forefront of our work in this alliance and use the information to direct the research agenda. To educate clinicians, regulators, pharma, researchers about what it's like to have overgrowth intellectual disability disorders. And hopefully this will lead to more efficient and meaningful drug development, decrease the amount of time it takes to bring treatments to our patients, and make those treatments relevant.
That's right. So like we said, we're stronger together. So in April last month, we launched our first meeting, and we had a virtual meet and greet. We realized we're stronger together, and this is our scope here. So the Overgrowth Syndrome Alliance, or OSA, we aim to align interests and resources of overgrowth syndrome patient organizations to galvanize research for treatment and cures. It just benefits, it just makes sense. If one of us progress, we all do. All organizations have strengths, and if we work together, we benefit. More meaningful data helps us set us up for success. We had our first, some patient organizations are new and they're solely focused on just patient support. Some don't have research networks yet. Some have really established research networks. We don't all have the same resources, but if we share those resources, we can collaborate and create a bigger voice. We can all tell you the same thing. We all have what our patients struggle with. We all have our patients focused. They're our number one priority. And we all want the best quality of life for all of our children and patients. We want to bring together scientists. So many scientists that study Milan syndrome and TBRS are also interested in several overgrowth syndromes. So if we gather these scientists together, present our priorities, provide a venue for collaborating resources and sharing research tools like patient samples, registry data, IPSCs, we can really drive research forward.
So our next steps for the Overgrowth Syndrome Alliance is our next meeting, we're going to have a virtual showcase of all of our organizations and where we are, what our strengths are, where our research is at. And then we'll collect our patient priorities with the patient priority survey, and then gather researchers who are interested in overgrowth together. Start to meet with the various research network members one-on-one and tell them about what we're doing and what our goals are. And in 2023, we're going to organize the first Overgrowth Syndrome Alliance conference and just see where this takes us. We want to increase the diagnosis of people with overgrowth syndromes and find them so they can get proper surveillance and also work on treatment development, like Kimmy said, identifying overlapping pathways. You never know what's going to be in the future. Maybe a center of excellence, a basket trial, but we're excited to see where it takes us.
Thank you both. Does this work? So, I just wanted to say, I've seen a number of patient diseases where there are fractured organizations.
We can't hear.
You can't
The mic.
There we go.
Okay. Can you hear now?
Yes.
I really wanted to say I think what the two of you are doing is great. I've seen a number of diseases where there are fractured patient organizations that are trying to address the same disease, and that doesn't help patients at all. And the fact that you're collaborating across diseases, I think, is wonderful. I also wanted to comment on what Shauna was discussing in her presentation about all of the different overgrowth diseases and how there may be different named diseases, some with the same gene, but many with the same pathway. And you also mentioned, Shauna, about natural history studies. And I wanted to ask you if you could comment on how maybe having multiple organizations collaborate on developing either integrated or sharing data in the natural history area, how that might benefit science and patients.
Sure, yeah. So when we're developing outcome measures, it's very important to be able to look at a particular condition in a variety of different ways. And I think I mentioned it at the very end there that people can measure things in an exam room, but it doesn't necessarily translate to something that's impactful to a patient's life and or their quality of life. And so when even going to an FDA to approach a clinical trial or a medication, they're going to want quality of life measures. They're going to want all of these different things in place to see, okay, well, you can measure, if someone has a reflex or not, but what does that do in the outside world landscape on their day-to-day life? How is that important? And so it's really working with patient advocacy groups that helps drive those types of outcome measures forward. And patient contact registries are a very powerful tool for that, where questionnaires and things can be developed, and you get hundreds or in sometimes thousands of responses very quickly, and that's very powerful for both patients and researchers and on other groups. So while it doesn't seem like exciting or super impactful, and research always feels so slow, these types of outcome measures, ways of measuring change over time, way of capturing what symptoms are included in a particular disease and what aren't, those are very important for the advancements of clinical trials.
Thanks. I have a number of other things I wanted to ask the three of you, but I know we only have a couple of minutes left until the break. So I wanted to see, does anyone in the audience have questions for any of the speakers? Well, I guess not. So, anyway, so my second question really is for Kim, and going further on the collaboration. I know that that you're participating in RareX, and I wondered if you could comment on what RareX is doing with neurological diseases and how that will benefit patients.
Yeah, absolutely. And just like you had asked about the natural history studies and having it go across diseases or disorders, that's what RareX is doing. It's a searchable database. So there's multiple organizations being that are part of RareX, and we were really excited to join it. So it's such an incredible force. It is new, but eventually RareX will have a dashboard for each organization where you could see what data is developing. How many people with Merlan syndrome, let's say, have seizures or a vision impairment? So they'll be able to put information in and get information out, which is really a huge incentive for families to get that feedback back. And what RareX is, for anyone that's not familiar with it, it is a database that has standardized questionnaires, and it is going to be searchable for researchers. So a researcher can come and choose a symptom, let's just say Chiari malformation. And they'll be able to put that in, and it won't just bring up Chiari malformation for Milan syndrome. It will bring up Chiari malformation across all diseases that are in that database. So it really is, it's such a it's just so versatile for researchers and Milan syndrome and other disorders that are using the platform. It puts us out there for more accurate research by symptoms.
Okay, well, I'm sorry. Oh, we have a question here.
I wanted to understand better the funding. If you guys are doing basic research in the field, what's the funding landscape for rare diseases?
Hi.
Anyway,
Tough.
the funding landscape. I'm interested to know. I work with the NIH, and a lot of the money usually goes to oncology, right? But when you're doing it for rare diseases, how does that look? And does it vary by countries? Are more countries more active in funding research?
Well, I can start and say that usually funding for rare genetic diseases is really tough and very tight. So in our case, we had to maneuver some of the funds and ask the agencies because we discovered, as I said, in our case, we were working on a tumor suppressor gene on oncology, and we discovered that our mice that we modeled tend to have seizures. And this coincided exactly with the fact that there were patients just reported to have seizures. And we had to ask for an allowance that we can go more in depth into this and find. And this was just the start. So we rely a lot on patients, on foundations to support our funds. There's not much, at least in my place in the country, to support rare genetic disease, very slim opportunities. And this is a big issue. This is really a very sad story here when it comes to driving the research. We do the basic research, and I just want to emphasize that this is very important and critical for us to move forward with therapeutics. If we do not understand how the genes are function in the right cells, for example, in our case in neurological diseases in the neurons or in the brain, then we will not move fast with translating this to something that can be coming up as a therapy. So we need the funds for understanding the basic mechanisms to establish these models and then translate this into therapeutics. So maybe.
So on my side, what I've seen, I worked at the NIH. There were extramural grants that people could apply for. And there was a big movement when I was training there to do sort of this bench to the bedside approach. So trying to get away from research that would not necessarily translate into population impact. And on top of that, whenever there's a particular research, also private funding through patient association groups and other sources can help. And once that moves into the private sector, then the funding comes from biotech industry itself. So it's multiple layers. There's some very early on funding, I would say, and then it moves closer to sort of like being part of a commercial product, and then different source of funding move into place. That's what I see.
Yeah, just a couple comments. All right. So thank you so much, Ellen, for, okay, am I moving to this one? Okay. Thank you so much, Ellen, for moderating and for our speakers today. I will have to say Dr. Akilan won the distance traveled to come to the fair. He came from Israel, so thank you. And like it was being said today, there's just so many ways that we can accelerate research for the over 10,000 rare diseases. And definitely finding pathways between rare diseases and repurposing drugs are two ways that we can do that. I will say Born Here started as a support group, and now we study 15 rare diseases, so it's really exciting. I also want to just quickly thank our sponsors again, Seattle Children's Research Institute, Pfizer, Pharma, Illumina, Vertex, Ultragenics, Alexion, Mazi Therapeutics, Regeneron, Decimal Consulting Groups, Born Here Research Foundation, and the Northwest Rare Disease Coalition. We couldn't do this without you. And now we're going to go on a break, but before we take a break, there's a poem written by a colleague here. I don't know if she's here, Anna Holt. She wrote it for rare disease patients, and then we turned it into a song from a musician. Oh, there she is. Can you stand up, Annie, please? And we gave it to a physician in Canada who turned it into a song, and we're going to make some rare disease videos, and this will be the song, the theme song for those videos. So we're excited to share that with you really quick before the break, and then you can go. Oh, I have to hit next. I'm sorry. Okay, there you go. Wait, did I go the wrong way? Did I go the wrong way? No. Oh, okay. Where am I going? Am I going the wrong way? Okay, I got this. Wait, is that it? Okay, no. No? Where am I going? Did I go the wrong way? Where's it going? Uh-oh. See, it's like all going different directions. Okay, here it is. So the poem that she wrote is called My Rare Wish, and I think some of us families can relate to this. So. I've been singing this song a lot at home, so my kids tell me to stop because it's catchy.
For the first time, I love that you are mine. How could we ever know the changes that we face and so? Teach us how to live with grace, to be strong in the face of tough times. I promise to fight hard for you. My wishes for your differences too. Shine through. You have changed my life for the better. How sweet it was that you chose me. I would argue I chose you, however. Now it's time to be a light. Let your journey begin.
Thank you guys so much. And guess what? You get to hear more songs from kids too after this that from Singing Me a Story. Some kids wrote some songs with medical complications in their siblings. So as you go out on the break, you will hear them too. Thank you, Annie, for that beautiful song.